Welcome back to part 2 of episode 10. In this episode, we're going to take a closer look at the auditory system. Here, we're first going to take a look at the brainstem nuclei, then the middle geniculate nucleus, then the primary auditory cortex, and finally on to higher cortical areas. After that, we're going to discuss how the auditory information is processed in the auditory system. Brainstem nuclei in the auditory pathway. This is a picture of all the brainstem nuclei that we are going to discuss. In the beginning, we have the cochlear nuclei. These nuclei receive information from the auditory vestibular nerve, which carries information out from the cochlea. There are two cochlear nuclei on each side, the dorsal cochlear nuclei and the ventral cochlear nuclei. Going on from this, we can see that some axons are ascending. Some of them cross over in a place called the trapezoid body, which is in the ventral part of the pons. Some remain ipsilateral and go up in the same side. Going up at the rostral medulla and caudal pons, we have the superior olivary nuclei, or in short, the superior olives. These nuclei send axons up through the lateral lemniscus. As we have mentioned in the episode of neuroanatomy, a lemniscus is a ribbon-like structure that goes all the way through the brainstem. Many axons take a stop at the nucleus of lateral lemniscus. Some axons just skip this stop and just go up directly. And inside the midbrain, we reach the inferior colliculi. This is the inferior colliculi and it sends axons up to the thalamus. Here is the map. Out from the cochlea, we go through the auditory nerve to the cochlear nucleus. The cochlear nuclei then bilaterally projects to the superior olive, which goes through the nucleus of lateral lemniscus and reaches the inferior colliculi. Now, up to the thalamus. This is the picture of the thalamus that we've also seen in the visual system episode. The medial geniculate body is posterior and ventral in the thalamus. It's here, just medial to the lateral geniculate body. We can see this through the similarity of their names, lateral geniculate body and medial geniculate body. In this map, we have now reached the MGN. Now it's question time. Please put the following structures in the order of information flow. These structures are nucleus of lateral lemniscus, inferior colliculi, MGN, superior olive, cochlear nuclei, and lateral lemniscus. Please take a few moments to arrange them by yourself. This is the answer. The first one is the cochlear nuclei, followed by the superior olive, the lateral lemniscus, the nucleus of lateral lemniscus, the inferior colliculi, and at the end, the medial geniculate nucleus. Now it's time to get to the primary auditory cortex. In terms of location, the primary auditory cortex, which is abbreviated A1, is on the superior side of the temporal lobe, here, somewhere near your eyes. It is the Brodmann's area 41 and 42. In terms of functions, one of the important functions in the primary auditory cortex is tonotopy. As we have mentioned about the cochlea, there is a tonotopy inside the cochlea. Similarly, there is a tonotopy inside the primary auditory cortex. We could introduce a concept called the characteristic frequency of a cell. This means the frequency of sound that stimulates the cell to a greatest extent. In the primary auditory cortex, there are isofrequency bands. In these bands, all the cells have similar characteristic frequencies. If we look at the most rostral part of the primary auditory cortex, we can see a band of cells that are most sensitive to low-frequency sounds. These correspond to the apex of the cochlea. Going back caudally, we see bands that are sensitive to higher frequencies, and these correspond to the base of the cochlea. Thus, tonotopy is preserved in the cortex as well, just like retinotopy is preserved in the visual cortex. Some other important information that are processed in this piece of cortex are, for example, intensity. There are intensity-tuned cells which have an optimum intensity that stimulates them to the most, just like the characteristic frequency for some cells. There are also cells that are responsive to specific types of sound, for example, clicks, bursts, animal sounds, voices, etc. In terms of information from both sides, there are columns of cells that have similar binaural interactions. Binaural means from the two ears. Therefore, if there is unilateral damage to the auditory cortex, the hearing will still be nearly intact. There will only be an impairment in an inability to localize the sound source. This could be contrasted with the visual cortex, where there are ocular dominance columns, but these are only serving one side of the visual field. The left and the right are very clearly segregated. For example, if we damage the left primary visual cortex, then there will be a loss of the right visual field. And another function of the primary auditory cortex is downward feedback. Just like the visual cortex feeding back to the LGN, the primary auditory cortex also feeds back to the MGN and other brainstem structures. Now that we have reached the primary sensory cortex, it's time to go on to the higher level cortices. One of the most important higher auditory area is called the vernix area. One of the most important higher auditory areas is the vernix area. The vernix area is here, adjacent to the primary auditory cortex. It is labeled Brodmann's area 22. Vernix area is in charge of the interpretation of spoken words. If there is a damage to this area, then the patient would likely have vernix aphasia. This is an aphasia where the patient is able to speak very fluently, but he or she cannot speak anything that makes sense, and he or she will have some trouble comprehending others' speech. In terms of other higher cortical areas, there is some evidence that supports a hypothesis of two streams of information, just like the visual cortices. On the dorsal auditory stream, we may have determination of localization. In other words, these cortical areas serve to help us localize the sound source. In the ventral auditory stream, similarly, we have recognition and comprehension functions. This unlocks all the parts in our map of the auditory system. Please take a closer look at this. This is our whole auditory system. Question time. Please select the correct statements. A. Damage to the left V1 will cause vision loss in the right visual field, and a similar damage to the left A1 will cause hearing loss in the right ear. B. A1 is in the occipital lobe, while V1 is in the temporal lobe. C. There is some evidence that suggests two streams of auditory information processing, similar to the streams of visual information. The correct answer is C. For A, damage to the left V1 will indeed cause vision loss in the right visual field. However, similar damage to the left A1 will not cause hearing loss in the right ear. In fact, the hearing will be almost intact. For B, we have gotten it completely wrong in the statement. A1 is actually in the temporal lobe, while V1 is actually in the occipital lobe. And for C, this is true. There is some evidence that suggests two streams of auditory information processing. Now on to information processing. Firstly, we would like to know about loudness and frequency. How are these two essential information processed in the brain? Loudness is the intensity of the sound stimulus. It can be measured by the firing frequency of neurons, as well as the number of neurons that are activated. This is fairly simple. Frequency is a little bit more complex. At a low frequency, there is a concept called phase locking. At every cycle of the sound wave, we can lock it to an action potential. In other words, the hair cell fires at every cycle. Since the frequency is not very high, the action potentials can keep up with the sound wave. When the frequency is intermediate, not very high but not very low, we can also use phase locking. But sometimes the action potential misses a cycle. In this situation, we rely on a mechanism called volley principle. Think of a volley of artillery in real life. There are a lot of artillery that are firing at the same time. It is similar in the auditory system. Multiple neurons are locked onto the phase of the sound. However, one cell does not cover every cycle. Therefore, we take the input of multiple neurons to ensure that at every cycle there is at least one neuron that fires. In other words, the different neurons can cover the cycles that the others have not covered. So phase locking is a very useful thing in determining frequency at low and intermediate frequencies. When the frequency becomes a little bit higher, however, then we rely more and more on tonotropy. This is because phase locking sometimes fails. The neurons are now unable to lock onto the specific stage of the sound wave. Therefore, we use tonotropy. As we have mentioned in the last part of this episode, there is tonotropy in the cochlea. And as we have mentioned in this part, that tonotropy is preserved into the primary auditory cortex. This tonotropy can help us determine the frequency when it is relatively intermediate or high. Now, on to a little bit more complex information about sound localization. For sound localization, we would like to know where the sound is coming from. For a human, this has two cases. Firstly, we need to know the horizontal direction. The horizontal direction can be determined in two ways, different cases for low frequency and higher frequency. For low frequency, we usually rely on the interaural time delay. The word interaural means between the ears. So, for example, if the sound source is directly ahead of you, then your two ears will receive the sound wave at the same time. If the sound source is not directly ahead of you, then there will be an interaural time difference. If the sound source is directly next to you, then the interaural time delay will be greatest. The time delay is processed in part by the superior olive. Here, the green circle in the middle represents the superior olive. The blue axons are from one ear and the brown axons are from the other ear. If the sound source is closer to the blue ear, then the action potential will travel first from the blue ear, then from the brown ear. This creates a time delay for all three neurons in this superior olive, and these neurons can process this delay and tell us where the sound is coming from. At higher frequency, however, we meet a difficulty in interaural time delay. Since the frequency is not phase locked now, we cannot calculate the interaural time delay very accurately. In this case, we can use the interaural intensity difference. What's that? Well, if a sound comes to us, then our head will block part of the sound, which means that the sound is attenuated in a shadow region of the head. If the sound source is directly in front of us, then the shadow will be directly behind us. The two ears will have no interaural intensity difference. If, however, the sound source is to the side, then one ear would be in the shadow, and it will have a smaller intensity for the sound. This is the case where the ear is in the middle of the shadow and the intensity difference is the highest. In the vertical case, we determine the sound location via the delay between original sound and the sound reflected by auricles. Let's take a look at this ear here. If the sound source is here, the waves can be transmitted by air directly into the ear. However, it can also bounce off the auricle and reflect into the ear. There is a time difference, a delay between these two types of sounds, and therefore we can calculate where it is, whether it's high or low, or whether it's just beside our ear. In animals, this is somewhat different. As you may have observed with your pet dog or cat, their ears can sometimes turn around, and this provides them with more freedom and more capability of determining the source of the sound. That ends our discussion about information processing. It's a final question time. Please briefly describe how do we carry out the following functions. A. Frequency determination when frequency is low. B. Frequency determination when it is high. C. Horizontal sound localization at low frequency. And D. Horizontal sound localization at high frequency. Please take a few moments to review this. Here we go. For frequency determination when frequency is low, we use mostly phase locking. B. For frequency determination when frequency is high, we use mostly tonotropy because phase locking is now failing. C. Horizontal sound localization at low frequency depends on the interaural time delay. And D. Horizontal sound localization at high frequency depends on the interaural intensity difference. That's all for the auditory system. In this episode, we learned about the pathway in the brainstem from the cochlea to the cochlear nuclei, then all the way up. And then we got to the middle geniculate nucleus in the thalamus. Then we went to the primary auditory cortex in the temporal lobe and reached higher cortical areas. Finally, we discussed information processing, including the frequency, the loudness, and the sound localization. That's all for the auditory system. Looking forward to seeing you again in the next system for sense and perception.